also know that we don't think that Isha had any intention of going, but we know that Isha's family would have been gutted if she didn't get that automatic admission. But it's our formulas that decide who's the top 10%.
So it's like people get
So we
counted.
give her this big boom.
Again, it's auto admit that have no intention of going. So it's like they have to admit more.
It's just, it's strange. Like the whole grade weighting thing has a profound impact on that.
Again, it's just perpetuating or exacerbating the whole notion that like you are your grades, which isn't like
Yeah.
true. And also, I don't think like dumb a person down, especially going to such a like college to present to see what you did for all your like 7%, your like doesn't make sense.
How did your parents get into college?
Into college. So both my parents I think were first gen. They went to college in Nigeria, and my dad for petroleum engineering, and my mom for education.
So my question is, how did they get into, so, like, you know, in India, they take a big test and the score on the test determines, like, choices.
Like testing, standardized testing, stuff. I don't know if it's called like GCSE, I think it's high school, but stuff like
Yeah,
that just gets used.
Okay. And so it is, like, absolutely the condensing someone into a singular number.
Yeah.
And, like, who cares? So it is the culture from which you were trying to
Just read from.
escape. Exactly.
But it's probably not gonna happen because med school is numbers.
I think there are choices. I think there are, like, a rich variety of choices. And I think that you are up to that challenge, you know? I think that people limit themselves because it's another place where everyone's heard of Harvard Med School. I feel like I don't have enough slides there, so I was looking for the other set of slides here, but I'll talk to you because you are all staring at me like it's my turn now. Hi. I have a witty acronym for you. This is something I forgot to tell your class that I told the other class.
The Tracy?
What?
The protein acronym, Tracy?
I don't know that. What's that?
Oh, that's for bio. Transport?
That's like the active and passive.
I was thinking of like the first, oh, okay, never mind.
Wait, what is Tracy?
It's a
But what is Tracy?
What kind of
person.
there are across the membrane, right? So like active.
What are the letters? What does it mean?
It is,
I haven't heard that.
we're just bio.
An example of proteins or a role of proteins is transport. An example of a transport protein might be ATP synthase, which transports H plus ions across the cell membrane. You may have other examples, but you know, he's my favorite. What are some other things that proteins do? Enzymes. My favorite enzyme is peroxidase because I think that some of you have done some labs with it. Catalase is also very popular.
Do they like the genetic material stuff? It's very biocentric. How would you describe what hemoglobin does? Is that transport again?
Yeah, I think I would make that transport. That's fair. I'm trying to think of in my like limited acronym here where else it would go. I don't have anywhere else for it to go. I mean, this is like 5 more proteins than you thought I could name. So like I fear the day when Margaret has to list like why do we have proteins as humans, and so she needs to come up with some function things. And I think that Amarachi could pull this out of her behind because she's in a biology class and she would just be like, let me think of some proteins I know the names of and like make something up. But I think the rest of us need like a somewhat offensive acronym to just make sure that we have some things going.
It could be time.
I'm working on it.
Is there more stuff?
Okay? I'll give you the H if you give me the thing. It's not hit me, don't worry, it's more offensive. So hormones are not proteins, but proteins are hormones. So there are some fatty acids that serve as hormones. There are some proteins that serve as hormones. So hormones is a large class of things, and proteins make up a portion of that class. Hormones are just messengers, right?
Is hit me the whole
Men?
No. Again, it's offensive. It's a dirty word. It's easy for Margaret to remember.
What about the ones that that have like collagen and stuff?
structure. Wait.
Oh!
It is!
Oh, yeah.
I did not say that. I didn't say it as a joke.
And yet here we are.
Wait, I thought it was gonna be something after the M. There's like a space.
I didn't know if I was gonna, like, there are two E's.
Oh.
They put a C on it in the other class. If you have something you want me to put a C on, feel free to add more. This is more than enough. I legitimately have no recollection. What? Maybe?
Oh, there's more.
No, like this is energy. You can eat protein. So if you look at Boris, who sits there in that class, you can imagine that he's just sitting there on a keto diet, like ingesting just large quantities of protein and teaching his body to burn that protein as an energy source. You don't picture Boris just being a little engine of protein. And so all of us, it doesn't really matter what the protein is, can eventually find a mechanism for breaking it down. It's probably not best. It has some consequences for the kidneys that some of you recently dissected. All of these proteins, to my knowledge, are globular, while this one is the opposite, fibrous. Fibrous?
Yeah. Thank you.
I was confident that I was writing, and I was like, ooh, poopy.
What about myosin?
You want myosin and actin to be fibrous?
Okay.
It's being looked up in real time. The question is, are myosin and actin fibrous or globular?
They're fibers. Myosin fibers.
They are both fibrous.
There you go, Bracken. I just picture that little head marching along, the little Z lines. It's one of my favorite biology classes, everyone. Not yours?
What's the heavy side? Muscle and biomuscle are like kind of
Because it has to do something.
It is pretty like understanding heavy. Like you can't really just memorize it. It has to make sense.
Yeah.
For you to like.
I really enjoyed that. You guys, I don't think of your biology career as having like highlights. I think you go like very often and just kind of learn. But I think like the parts I enjoy, and I have completely no recognition of the parts I hated. Right, like meiosis, like all I can tell you is you PMAT twice. And like it means essentially nothing to me. Yes, PMAT, like, but that's my level of understanding of those different phases. Like it's just a sheer, like, I don't think I care, and I'm sorry I don't care. Really, I love the genetics. The epistatic genes? You don't love that?
I mean, genetics like in and of itself as a concept is cool, but I'm very tired of like what's called pedigree chart.
Oh, yeah.
I don't like
Find out who has lived
Yeah,
around.
yeah,
who's
yeah.
not.
I don't like this stuff.
I know there's a feeling, but like some of them just not pretty.
Punnett squares are fun though.
Like, who doesn't love this story of Labrador retrievers and the epistatic gene? See, if you don't have a big E, you can't be a brown or black Labrador retriever. You'll be a deeper, even if you have the big B.
See, stop that. We have like recessive menopause.
I got the baldness thing. See, baldness is dominant in men, but if you have both of the recessive genes and you're a girl, you get it.
You're just superior or you're bald.
No, you're bald. So if you're X little b, you have girl balding. If you're X big B, X Y, you're a bald man.
Is there a reason why it's not from my father?
Because you, like, it's recessive.
Is less likely.
I know, but like, okay, I guess.
I don't know. And like the sickle cell stuff, there's so many good stories. See, everyone likes the sickle cell one. Even Mohab? You still coming down for your birthday?
I mean, yeah, that's good.
Do you have milk on your bones?
In like 30 years.
You have to look at your mommy.
My mom?
Yes, your mom's father is the determiner.
My dad is.
I don't know who my mom's father was.
Well, then it'll just be a surprise. Should you have a child? It'll just be like, who knows?
Is male pattern baldness like a complete inability to grow hair on your head, or just like the majority?
It just makes us younger.
No, it's just like,
That's why like some
Yeah, it's
people
not
in their
alopecia.
20s will
Yeah.
like bald.
isn't that like?
20s?
Yeah, it like happens really early.
Yeah, my dad was bald really young.
No, my dad was bald for a long
Isn't
time,
like
but I don't know.
some kind of like hormone or something?
See, that's the thing. There's natural factors. It's like a hereditary factor. You know, it's people start getting it.
That makes sense. It is crazy, but it does make sense,
It
right?
makes sense, but it's just like.
And then like there's the testosterone receptors, and like some people can't turn them on, so even though they have testes, they can't tell that they're making testosterone, so they develop female genitals even though they have a Y chromosome, because they don't, like, yeah, like, I don't know, I really liked all the goofy stories that don't matter to anyone but the teacher who's like off in their own dream world.
Which is
I think that's the worst part though.
Okay, so we talked about functions. We were in the throes of chromatography last time. You guys were loving it. You guys were like, more, more. And so today we're going to talk about, I don't know, we're going to talk about primary, secondary, tertiary, quaternary structure.
Joy.
Okay, Sophia's going to be on the list of people. Sophia will play the role of P today. No, P for Pumi. I pick on him a lot, right? He chose a major in biochemistry at least one of the schools he applied to. We are waiting for an email to arrive. So last time we talked, we, I believe, talked about taking 2 amino acids and making them into a dipeptide. Do you remember that? Good, because I think we did it, but I have like no idea. And so we said that if you took serine and alanine, you could make, you know, not 1, but 2 different dipeptides.
Well, I thought I told you when I got any.
Oh. Oh my goodness. I believe this is serine. I'm confident this is alanine because I draw it like every day and all the time. It's one of the ones I know. And so what are the 2 dipeptides that I can make that contain those 2 amino acids? It's not a trick question, everybody.
Serine and alanine.
Yes, I have serine and alanine, and I want to make a dipeptide. I claim I can make 2.
Are you asking what part of this?
That's what I'm asking. So Karthika says I can add them like this, or I can add them like that. And so we always read proteins from the N-terminus, And so it would be a reasonable question, this is a drawing heavy unit, for them to say draw the two dipeptides. And so this problem didn't take me a whole lot of time to think about, but maybe it did take me a minute to draw this all out. And maybe you guys are better at it than I am. But I'm showing you that peptide linkage, I am showing you that the order of the R groups has swapped, and I am reminding you that as good little biologists, we always read from the N side to the C side, from the N terminus to the C terminus, y'all might say. That's what you say, right? N terminus, C terminus? We read them that way because that's the way they grow in ribosomes. Like when the tRNA comes in and puts another little amino acid on the protein it's making, it always adds to the C side. No. I don't have to do that till I'm H shell. She's talking about 5' and 3' ends of DNA, and I only have to deal with that when we're in the HL part. I'll like learn how to count to 5 and understand why it's 5 and 3. And you will judge me and I'll be fine with it. I mean, on a sugar, like, why is it 2-deoxyribose? Because it's carbon number 2. Where is carbon number 2 in the ribose? Why is it 3' to 5'? What am I counting and where is the 5? That's what I mean. I don't know. So I have two versions of the same slides. I just felt like there were some more things in here that might be better. So I was trying something different for you. So here I've taken a polypeptide. It's a whole lot of peptides. You can see that there are R groups coming along, different ones, 4 of them. How many waters would I give off in this process? For a dipeptide, I'll take that. 1 for the dipeptide here. There have been at least 3 lost because he's answering one question, you're answering the other. So just don't forget the water. So this is a real kind of question they might ask you if you have 3 amino acids, glutamic acid, histidine, and glycine. How many unique tripeptides could you make that contain each of those 3 amino acids? Six, 3 factorial. Everyone who's thinking 6, 3 factorial. Again, this is a question that comes up regularly and does not involve your encyclopedic knowledge of biology, which is good for me. But when we list this, do you notice that we start at the N terminus, we go to the C terminus, and we're just telling you which amino acids are there using their 3-letter acronym. Where would I get their 3-letter acronym? From the data booklet. I actually refer to that as the primary structure. So the primary structure is the listing from N to C of the amino acids in order. Primary structure. Every protein has a primary structure, and it's like not terribly interesting. You want me on those slides so you can take notes on this? I don't care. Just in case we need it. Anything you think I should add about primary structure? Like I don't know anything about it. It is really just the order of the amino acids. Secondary structure is where things get a little bit more exciting. I don't know, this picture is a whole lot better than the picture that's in your notes. But secondary structure relies on hydrogen bonding, but it's a really specific hydrogen bond. What you're going to notice is that each amino acid is forming that very long peptide bond, right? But this hydrogen is a hydrogen that is directly bonded to an oxygen, nitrogen, or hydrogen. And so it's got a pretty large partial positive charge. And somewhere else in this world is another piece of this peptide chain. And in that peptide chain, there's a carbonyl group that's got some lone pairs on a partially negative oxygen, and we can form a hydrogen bond between this guy's hydrogen and that guy's oxygen. So secondary structure is due to hydrogen bonding of the peptide backbone. And that's important because every protein has it. So we find these structural motifs in every protein. The two most common structural motifs we discuss are the alpha helix and the beta pleated sheet. In an alpha helix, we actually form like a corkscrew kind of structure, a helix, where there's 3 or 4 or 3.6, depending on how you count, amino acids in between each curve. And so I'm looking at the amine group, or the amide group, I guess, it's like 4 guys ahead of me, and I am making an interaction right there. I am making an interaction right there, and that's what determines the size of this helix. Like that's the tightest helix I can make to maintain those hydrogen bonds along the length of it. The other common structural motif that you'll see is known as a beta pleated sheet. In this beta pleated sheet, it's kind of the one I prefer, what we have is like a stretched out section of peptide that then comes to a kink. Kink is usually formed by a glycine residue, and then it comes back and doubles on itself. So this is the chain, and then it comes back and finds itself. But in between, what we can do is we can make hydrogen bonds between the NH and the C double bond O. So a beta pleated sheet and an alpha helix are common structural motifs that we'll see in anything. And so I'm not super good at naming proteins, but hopefully you named some for me a minute ago. That was a bad guess, apparently. Okay, it's fine. What you can see here are those alpha helices. You can see there's more to the shape of peroxidase, but this structural motif is a part of peroxidase. What if we did, I erased everything, hemoglobin. One more time, you're going to see a whole lot of alpha helices. Now, what are the green ribbons in your estimation? What? Oh, I don't think that's beta. That's a fine answer. What I believe it to be is parts of the protein that aren't well described by secondary structure. I think anytime we see something that fits our definition of alpha helix, like in the protein database or whatever, we put it in like this bull like ribbon form. And anytime we see a beta pleated sheet, just want a protein with a lot of it. What's a complicated? Let's just go to ATP synthase and see if it just makes my day. Apparently I guessed a little bit better. Look at that thing. Look at it. All the different colors, all of the different sections. Look at that, all of that alpha helix in there, but there's not a beta pleated sheet I can see to save my life. X-ray crystallography. X-ray crystallography. So they actually make crystals of the protein, and then they figure out where each and every atom is in that backbone, and then they render it. It is really hard. So in my day, that was the longest chemistry PhD, was X-ray crystallography. It took a solid 7 years. Solid 7. What'd you learn?
you like didn't mention that.
Oh, yeah. It's in chapter
That's how they have
21.
structured DNA.
Yeah, that's how they determine.
Yes, they look, yes, Rosalind Franklin had that in her drawer and Butthead went and stole it.
Yeah.
That's wild. Stealing people's
Scientists were
stuff.
scandalous. They would steal stuff.
It's because those people who are at the very top of the science game really want their Nobel Prizes and science awards or whatever.
Come on, does anyone have protein? Oh, here we go. Who knows what it is and who freaking cares?
What is that?
So this is another protein. I don't know what it is. I apologize. I mean, I care a little bit. It's clearly got something hanging out here in this active site, perhaps. Looks like it's doing some business. But you can see these blue stretches, and the blue stretches actually have arrows on them. That's telling you the direction of the protein chain in a beta-pleated sheet. So we point out the alpha helices, we point out the beta-pleated sheets, and then the parts that are in between that are folded by something more, something different, something perhaps we'll call tertiary structure, are just shown as skinny little ribbon. Like, we don't understand that from this picture.
It's tertiary.
That's gonna be tertiary structure that's folding it up but isn't the secondary structure. Does that make sense?
So which is the alpha helix,
Alpha helix is the red part.
the beta, and do we not see any beta pleated sheets?
Beta-pleated sheets are all of the blue part.
How are those beta pleated sheets? Like, you know how you?
Because they're like straight and parallel to each other. There's hydrogen bonding here.
Okay. Yeah,
Alpha is good, right?
You're not feeling that?
not really. I mean, like,
Okay, let me try this one, see if you like it. One, Mas Grande.
it's like, is it like alpha helices, betas, it comes back around, and then hydrogen bonds?
Alpha bends, the bend chain. That way it's spiral with the helix structure versus beta being straight.
Yes, so one is coiled up, and the reason that it coils up is because it can get all the hydrophilic R groups on the inside. And so that's why I think it's a structural protein. I don't think they're typically soluble in water. I think they like hide the R groups. And a beta-pleated sheet, I think, tends to expose the R groups a little bit more and tends to be a little bit more in a hydrophilic environment. But that is how you would look at the residues and try to make a decision, are you likely to be alpha or beta? So I think you guys can do that, but perhaps in a course that I'm not gonna teach or be in.
And the green stuff was the stuff like that we didn't like understand.
So it's not that we don't understand it, it's just that it doesn't fit our definition of either alpha or beta. And so there is something folding it, it's gonna be the tertiary structure that's governing there. I don't know, this one had like a really rockin' picture, but you all see that there's no big picture for me to look at.
What does the direction mean for the?
The direction is going from the end to the C terminus. And so my understanding of beta-pleated sheets until, I found some better websites and stared at them, was that they had to fold in on each other, that it required the U-turn, but it ends up that they can do it either in this or this conformation. I don't know how it would be different to someone who understood this stuff, but it clearly is not, doesn't have to be a fold on yourself.
Wait, what's an N and C terminus?
The N and C terminus, when we're drawing the amino acid, the N terminus is like the beginning of the peptide, and the C is where the peptide would continue growing if it was growing in you. Can you believe this stuff is going on in you right now?
No, not really.
You're making proteins.
Before when I felt first, I thought chicken, and now I think about.
That's not healthy.
Crazy. Okay, so this is another one with lots of beta pleated sheets. Does this one look more parallel, Mohab? Does it look more pleasing to your eyeballs? Huh?
They look like green beans.
Wait, so why are we, like, proteins don't die? Like, but they're not alive. But they can die.
They should get digested.
What about
Imagine every single cell in the body was sentient.
Oh my gosh. So what is your question?
I don't think
I'm just
anything.
confused, like, why would you save them? I didn't think they died.
They would start rebelling, and you just die.
They want to go on their own. They want to be like 10%.
Because you're making new cells. So like when you apoptose, I don't know what happens to the proteins. I don't know that the proteins can like go into the next cell and be like, yo, I'm ready to work. I assume they have to be like rebuilt out of protein soup.
That makes sense.
Did he
always like talking, like, what does the Golgi apparatus do?
You could just be like,
How many people get into a theoretical?
So, right, it modifies and packages, so I feel like that piece must involve some sort of building something that plugs in.
How is,
I don't know.
carbon and other stuff?
There's so many ribosomes, like we talk about all the time, every cell. The rougher you are.
What if,
If y'all ever come back and like actually understand how all of this goes on, I'd really like to know.
I was just saying, I don't understand how all of this stuff is like happening like everywhere but like in your brain, and then some of that is like you being sentient or you being conscious. Like how does that make any sense at all? I don't know sense.
what if?
So for the thing for Preston last night, they brought in some radio medicine speaker. And so he was talking about labeling glucose, so they make radioactive glucose, and that's how they decide if chemotherapy is working, because cancer cells eat more than other cells, and so they can watch where the glucose is going, and they can watch if less glucose is going there and say, our chemotherapy is treating your cancer. Oh my God. Like, in the scheme of like, how does this make us alive, then you have like parts of you wish were living worse, and like, how do we modulate that? Then how many pounds of you are other living organisms?
Like we're like, isn't it 10 times more bacteria than actually we're human? That's insane.
I think we got that information.
Writing number, writing.
She's doing a project for Pat though, everyone.
I'm sure.
She's not a good person. But Rainey's like here today.
Happy to be.
It's been a minute. So tertiary structure is the third layer of structure. It's not just the order, it's not the hydrogen bonding that can occur between the peptide backbones, but here what we're gonna look at is the actual R groups that we've got. Yeah, I keep getting email, I'm sorry, I'll shut it down. It's just gonna wake you up, like it's just a periodic, random, so you don't get used to it, really loud noise.
That's literally me and Mohammed were saying yesterday, you remind us of our seventh grade science teacher. It's perfect. There's one time that people weren't like paying attention, or people were just like kind of like brain fog, and she just walked to the textbook and just dropped it on the ground. With these loud my little
do you know Miss Murphy?
Of course.
Because she's always in the middle sometimes.
She taught Joseph.
I didn't.
she left, I think.
I mean,
She went to Hungary to teach at an international school.
I didn't know like high school and middle school teachers like interact.
Yeah.
I mean, because I had a kid in it, I might know more about it than some people, but we have meetings with them periodically. We have like a hot date with them on Feb 20 to talk about science.
Hot science date.
I think because Colleen Talfest has her daughter here, she's pretty invested in what high school science looks like. Was Talfest not here for you?
Colin,
Okay.
Colin was like the one in our grade.
Oh, Colin, that's right. I forgot. Like, I didn't know that Madeline was going to go here when Colin left.
Where is Colin?
Taylor, I think.
Oh.
Taylor.
Baylor.
Not Baylor. Subtle, yet different. Okay, so there are going to be a few different interactions in tertiary structure that you're going to be responsible for. So this is interactions of the residues or interactions of the R groups. And to me, this goes back to our discussion of chromatography or the gel electrophoresis, where we're looking at properties of those R groups and deciding like why they might move more or less. So you could imagine that you have a class of R groups. It's going to be valine, leucine, isoleucine, glycine. And if you check those out in your data booklet that was carefully provided for you in table 33, what you're going to find is that all of those R's are made out of alkyls. And so if I'm an alkyl, what kind of intermolecular force do I have? So all of these guys are only going to be able to engage in London dispersion forces. And so that's an easy answer because there's only one, I don't have to worry about it. And you can see an example of that right here. On the bottom, we have leucine. On the top, we have tryptophan. And so we have a big, juicy hydrocarbon there with that benzene. We have this butyl chain that's sticking out. This interaction is just an induced dipole interaction. So it's relatively weak. On the other hand, arginine had a whole bunch of those NH2 groups, and glutamic acid had a coup. So glutamic acid and aspartic acids have coups. And there are a few guys that have some NH2s, but my favorite are lysine and arginine. And these guys can actually interact in two ways. The more common way is that this protein is in some sort of a pH. We don't know what the pH is, but we learned even for a humble amino acid that there was a real probability that the amino acid was going to find itself protonating itself, forming that Zwitter ion at kind of neutral-ish pHs. So depending on the pH that my protein is in, I could imagine that a glutamic acid somewhere is able to find a lysine somewhere else and give it a proton. So it's going to be an intermolecular hydrogen or proton transfer. But when that happens, we will have the carboxylate, and the ammonium, and these two things can form an ionic bond. So we actually get tertiary structure depending on the pH of the form of ionic bond. So that's what you see right here between the glutamic acid and the arginine. Is there a chemical reaction that's possible there? Do a Q and an amine do anything? So they can neutralize each other. That's what I tried to sort of show you here. But they can also perhaps make an ester. It seems conceivable you could make a covalent bond here. So that is what you see occurring between lysine and glutamic acid. Lysine and glutamic acid. They actually formed a peptide bond in between two R groups. So if we're wondering how we're going to fold up, we're going to have the ability for me to make a bond to Amarachi. That's going to make this whole loop of our protein change in shape. Because making bonds is always exothermic, it's going to make this system more stable.
Wait, I have a question.
Yeah.
So the thing you wrote on the board, are you saying that those 4 ones, they only track with other ones with LDFs?
Like their only mechanism of interaction is LDF. So we know that everyone can LDF,
Okay.
but like the only thing those four can do is LDF.
And then glutamic and aspartic, they interact with the food on an amino acid, and the other ones they
Okay, and so I'm trying to convince you that these in turn can make an ionic bond, depending on the pH. They can make a covalent bond. That biologists don't speak of. I don't know how common this is. Real questions there.
Water, but
Yeah. I just I don't know how much. So when we talk about why my hair's not curly, you are all going to say it's because I don't have a lot of cysteine linkages. Like the disulfide bridge is what we associate with curly hair. My proteins don't have a whole lot of cysteines because I have some pretty straight, boring hair. But if we imagine Alvar in the room, he's got perfect coils, he's got some serious cysteine going on in that hair. But that's also perhaps why his children are going to be likely to have curly hair, because it's clearly a protein that he is expressing that's making that hair curl. So that Amarachi doesn't judge me, I'm going to put LDF here. There's nothing to stop it from making LDF, but it can make these stronger things. And so I think it's interesting. Another type of interaction that we can have is going to be something like serine, which has an OH functionality. You guys are going to tell me there's a pKa associated with it, so maybe it could lose a proton and become like an acid-base thing. But on the whole, my expectation is that this is probably going to find something that hydrogen bonds to. Now, it can hydrogen bond to a glutamic acid. It can hydrogen bond to a lysine, but I have him in his own column because this is like the only interaction that's meaningful that serine does. Reasonable way of differentiating these things? And so my last category, I'll put cysteine over here by itself. We take a cysteine molecule here, we take a cysteine molecule somewhere else, and they form a disulfide bridge upon losing 2 units of H plus and 2 electrons. It's actually a reduction reaction. I imagine that this has something to do with how hair perms work. I imagine that somehow we're trying to reduce your hair. I imagine that's why there are rules about getting it wet and how long you keep it. And I don't know. They were looking up what you use to do it, but I don't know.
Can you develop more of those like disulfide bridges over time?
I don't know how you would start making a new protein.
Well, the only problem is like
Yes, like, that doesn't mean, like, tell me your
like
story.
my hair is like kind of weird right now, but like when I was like younger, it used to be like straight back.
Really?
But I never did anything. I just like lived my life.
There's
But like it's just kind of like I mean I swam a lot like in middle school. I didn't do anything. Like, it just kind of like became like this. But it used to be like that this way when I was like 8, 9.
It's interesting. I don't know how people change over their lifetime. So we know that like you have operons that control like how much of some things that you express, right? And so we believe, I don't know who we are in this sense, so I'll say I believe, but that makes it sound like, I'm just making shit up. Maybe I am. That you're meant to process lactose until you're about 4 years old as a human, right? And after that, you're meant really to be lactose intolerant. There's no reason you'd be taking in milk at that point in a different world. Is that fair?
Yeah.
But we believe that you would no longer probably be expressing lactase, so I believe that you're meant to turn that off at some point in your life. It's not going to be super surprising that I had, I'm going to go with rainy blonde hair when I was a baby. Yes, and so as I've gotten older, my hair has gotten darker. I don't have a reason that my hair is going to get darker as I go older, but eventually it's going to turn gray. I don't know if that's also a switch in what proteins I'm able or not able to express. So there has to be a change over time, and like consult Operation Patho here.
I mean, it's just that like it wasn't as straight as it is now. Like when I was young.
Sure, but like, is this not a known thing that your body works differently as you age?
I had lots of curly hair.
Straighter and straighter?
Yeah.
Margaret?
My sister used to have ringlets, but then it stopped, and I didn't have, my hair didn't grow a lot when I was a kid, but then it just stayed like this. That I know of, nobody in my family has.
Margaret, your hair the same as it was when you were born? Imagine those little pictures of you.
I think so, yeah.
Your brother, same, straight, dark brown hair? So vanilla. Quinn, you got like a whole genetics experiment going on at your house.
I think it's the same.
I think the same way your hair would change as you grow. Like you said, it's because of the disulfide bonds or whatever. Like the majority of my hair. Actually, it's weird. Like my hair, like I kind of knocked out over like the course of like taking care of my hair and like switching from relaxed to natural. I like, and when I got into like my hair, the front is like super like kinky, and then the back is kind of like a bit looser, and then like here, like the nape of my neck is like a bit tighter than like Alvarez. Like it's
Okay.
a completely different texture, like all over my hair.
Fascinating.
And I don't know why. Like as a baby, like I obviously I had like a low cut
just, I don't know what biological mechanism is making your hair change, right? Like what are we getting out of it, right? Don't you wonder? Are you super light?
No, my hair is getting dark brown.
And it's getting lighter?
Yeah, my hair's like red now, but in all my baby pictures I had dark brown.
That's crazy.
My mom went the opposite. She went from like your color hair to a dark brown.
But like, I don't know, it didn't change when I was like young. It changed when I was in like middle school.
I don't know if puberty has anything to do with our hair.
Hormones.
Hormones.
Just puberty and apparently vitamins too. Somehow, but apparently that can also cause change.
What kind of vitamins do I need to take? I want better hair.
Biotin?
Okay.
No, biotin would make your hair grow fast.
It says insufficient vitamin D to make it grow badly.
The only thing that can really make your hair, the only thing that determines how fast your hair grows is your genetics, but things that like oils and stuff and biotin, like they just help with like hair
Texture.
length retention, which is what you see in the growth. So like if your hair is growing super fast, but it's also breaking off at the same rate, you're not gonna see any change. But like vitamins that like help strengthen the hair strand or whatever, like you'll see your hair actually.
I swear biology is fascinating. Y'all should really like take a magazine approach to that class because there's like there's every turn in something interesting. I understand that every class that you take you learn lots of stuff you don't want to know and it gets really hard, but super cool. Look, it's hemoglobin again. Wow. Hemoglobin is cool because it's actually 4 chains that have already folded. So each one of those chains has a primary structure that's telling me the order of the amino acids. Each one of those chains has secondary structure. You can see a kinky little alpha helix right there. Each one has tertiary structure. They folded together, but then 4 of them come together and make this more complicated hemoglobin. That is what tertiary, that is what quaternary structure is, sorry. Quaternary structure, 4 of them at a time. I didn't read anything good. Enzymes are really the focus of B7 from my point of view. That's when we're going to start doing math and start reviewing some of the joy of topic 16. You might remember that's when we had like a full reaction mechanism and we were able to write out a rate law for each of the steps and come together to understand the system as a whole. So you might also imagine, as people who have taken some coursework, that in addition to the traditional enzyme and substrate, there are some interlocutor, is that a word? Interlocutor, it sounds so fancy. There's some other stuff, see that's how I normally talk, there's some other stuff that can go on with enzymes. I don't know, like it just sounds like the right word. Sometimes when I'm writing your letters of recommendation, you know, because they're written now, I get a word in my head, I'm like, I've got to get this word into a letter of recommendation. Ebullient's one of my favorite. Such a good chemistry word, ebullience. Ebullience. So it comes from the notion of boiling. So it's someone who has like a bubbly personality, someone who's super like B-U-L-L-I-E-N-T, E-B-U-L. But it's where we get bouillon from, like soup bouillon, because we're gonna boil it to make soup. Huh?
All of us are just whatever that word is.
No. No, like, I mean, I'm sitting there typing and I'll get a word in my head and like I'll notice that I really want to get it. So interlocutor, like it's the word I feel like.
It is a word, but it means, it's like something to do with language or translation.
No, that's not the word I want.
It's a
Interloper?
What are you trying to say? Like
Other shit is what I'm trying to say, but I'm trying to say it with a fancy word. It's like 6 or 7 syllables, so y'all think I'm cultured.
Extraterrestrial.
What's your at all? I'll speak Latin, something I know even less about. Okay, I like interloper.
And others.
interloper.
And co.
A person who becomes involved in a place or situation where they are not wanted are considered not to belong. So that's the word I wanted. So I'm thinking that you guys know something about enzymes, even those of you who don't enjoy the class we talk about a lot right now. to me, this is what an enzyme does. An enzyme E binds the substrate S at its active site to make a complex enzyme substrate complex. The enzyme substrate complex then releases the substrate that it has converted into a product. So an example of this would be lactase taking on lactose, because biologists have no imagination. They make this special little thing, and here we get lactase ready to go again, and 2 monosaccharides. Y'all can tell me what they are, but I haven't looked it up.
Glucose and fructose.
Fructose?
Glucose and fructose.
Galactose. Fructose and glucose make sucrose. It's the only one I could have answered. Sometimes we talk about this in terms of a lock and key mechanism. We say that lactase has a perfect hole, and that perfect hole in lactase has exactly the right shape for lactose to fit into and break that stuff apart and make this. Audrey's like, no, that's not the current model. We now believe that there's an induced fit model. The induced fit model says that there's a special spot in lactase. As the lactose gets closer, the lactose changes its conformation, the enzyme's active site changes its conformation, and now suddenly they work together. It's the same model. But the induced fit model gives me something to think about. What it gives me to think about is, what if Emma's like, lactose is good, I make lactase. I want to know if I can use this same enzyme to break down maltose, which would give me glucose and glucose. If it's truly a lock and key, then maltose won't fit in the lock. And so we should get no product. But if it's truly an induced fit, then the expectation is that if I put a sugar that's pretty similar into that space, that the enzyme and the sugar might be able to arrange themselves in such a way to make it maybe not as good at cleaving this glycosidic linkage, but possible. And so if I had a bio-YA to write, which blissfully I don't, I would look to see if enzymes are good at doing other stuff. What is the relative efficacy on disaccharides? Because I can have a nice categorical variable, #biology, 5 disaccharides, let's go. How fast am I making glucose? Shove a strip in there, done. Good? Did you give me a 7? Okay. That's all I need. So my interloper, because you guys are all crazy English beans who know words better than me, is what you might call an inhibitor. And that inhibitor can do 2 things. The inhibitor can either go to the active site, blocking the substrate, that's competitive inhibition, or that inhibitor can go to the allosteric site and mess things up for everybody, that's non-competitive. There's also uncompetitive, because again, the biologists hate me. There's non and un, and I'm sorry.
What's the difference?
They're different.
Linguistically, they should be the same. The un and non are both
What is on, so they just don't exist?
I don't know. We'll see if we have to learn that. So we are going to think about the kinetics of this process. It's great. Kinetics. But for this unit, when we're just chilling in the SL, not going to worry about inhibitors, and we're just going to think about a really simple picture. I'm going to call my y-axis the rate of enzyming. You might call it the turnover rate, you might call it the reaction rate, you might call it the velocity. Again, I don't know what you call it. Feel free to yell out options.
rate of you should call it reaction.
Reaction rate.
And on this axis, I'm going to show you the substrate's concentration. As this is a mechanistic step, everyone in the room should be able to draw this picture. What is the order of the rate with respect to the substrate? Everyone in the room is drawing a straight line because it's first order process, but then Amarachi went, woo! That's supposed to be horizontal. Apologies. This reaction is first order until What?
Everything was plateau where
So
the
active
maximum
sites are
like amount of this the maximum amount of like enzymes that we can use up, so there's not much more substrate.
So when we run out of enzyme, we can no longer use up substrate, is what Amarachi said. It's absolutely true. In this unit, you're supposed to be familiar with this shape. In the next unit, we're going to isolate this point. That's going to be the rate that is halfway to that plateau that you described. We're going to look at the substrate concentration there. We're going to refer to that as the Michaelis constant. And we're going to learn how inhibitors might change the location of that particular point on the graph. It's really easy to spot it. Like, it's really easy to define. So that's coming. It's going to be a nice review of 6 and 7. But I think it's too much for now. But I was going to give your class the choice. So you have a quiz coming up at the end of next week on biochemistry. It's going to be on B2 because we're done with it. We're just trying to make this stretch out today, B2. And then it might be on B3, that's lipids. Or it could be on B7, that would be HL proteins. Now I've printed my lipid work, got my slides ready for lipids, like I'm ready to go. But if you think it makes more sense to like stick with this, if you think this would be a useful review for your mocks, although the content is harder, you could talk me into it. So do you want the easier stuff or the harder stuff? The harder stuff is like math, like rates and equilibria. So Sophia clearly doesn't want it because Sophia knows all the words for lipids. Like she's not scared of me saying this is a steroid. There are 4 rings. Let's call them alpha, beta, gamma, delta. I don't know what y'all call the rings. Would you call them anything? I don't know.
I don't know. Those never really come up on the test.
I need some more Mandarin speakers. One of the words for like cholesterol just has like a picture of 4 rings.
That's really cool.
We'll talk, Audrey. We'll make you my Mandarin speaker, okay? It's not just like it's a complicated story. Everyone's looking forward to me telling it. Okay, so you can think about that. Clearly, you don't want to think about it. But here's like a blast from the past. Here, my reactant was lactose. My products were glucose and galactose. Is this reaction endothermic or exothermic? Exothermic. There's net energy release from the reaction. But there's an activation energy, and the role of the enzyme is to lower that activation energy. That is how it speeds up the reaction. It provides an alternate pathway for lactose to cleave into those 2 monosaccharides. Happy blast from the past. Marcus not even writing this down. It's like enzymes I understand. I'm going to say again, the purpose of all of this option thing is to put it in a different context, but to teach you the same chemistry. Like my motivation is topic 16. Your motivation is to tell me about how things work. This is what we're talking about with lock and key, with the active fit model. That's why I changed these to be the slides for today because the other ones that I printed didn't have anything. So this is the substrate coming up to the enzyme. You can see that the enzyme changes its shape and the substrate somehow self-assemble. So both of these things conformed to each other to make that enzyme substrate complex, and then the product was able to come off and the enzyme is ready to do this again. So that's just a better picture than I'm capable of drawing with my hands, but hopefully an idea that makes sense. If we wanted to do the lock and key, the enzyme would always have exactly that shape. So when we're saying that the enzyme has a shape that is influenced by the substrate, that's what the induced fit model is trying to navigate. You drew this picture for me. Oh, this. So one of the ways that we can change the rate of enzyming, because you all liked that phrase, was by changing the substrate concentration. So we drew that picture, but we're going to draw 2 more pictures. They're already there, so we're just going to try to explain them. The first one is going to be how pH might affect this rate of enzyming. They call it enzyme activity. I'll just call it rate. So I believe that in your body you have 2 places where you use amylase. I believe that in your mouth, amylase breaks down starch and gives you that sweet, glorious taste of white bread or rice. Sweet. I believe after your pyloric valve, chyme is shoved out of your tummy into your duodenum.
Yeah.
Is that not?
No, yeah, I don't know.
I say duodenum, but I thought it was
I
duodenum.
say duodenum.
I say do it. Why did I say do
I mean, human.
Okay, I don't know. Into your intestine. Lord knows I don't know what I'm talking about. And sooner or later, a pancreas is going to be there, and it's going to be squirting sodium bicarbonate. That's the noise we make in our house when you eat something sweet to talk about the insulin that's being injected into your bloodstream. So
Why are you
something's
talking about
really,
that?
really sweet. My partner will start going.
Seriously?
His mom has the diabetes, so it's something he thinks about. And so my pancreas is currently not making insulin. My pancreas is currently making sodium bicarbonate, but the kind that left my stomach is really, really acidic. Right? And so the pancreas is going to have some amylase that's going to further break down starches that made it through my stomach. I don't know why I think my stomach's here. It's not, but whatever. Lower, mouth, stomach. Are we good with this? So I believe that my mouth has a pH of around 7. I don't know. Y'all can.
Good enough.
Sure.
And I believe that my chyme post sodium bicarb amylase is going to have a lower pH. Again, 4 is a completely made up number. Let's be clear. It's just got to be different and acidicer. How would the rate of these two versions of amylase look different given the fact that they're meant for different environments? Okay, so I think you guys are agreeing that I tried to choose where the optimal pH is. And that if I were to change the pH, that is if I were to spit into my duodenum, the amylase in my spit would not suddenly be able, I don't know, to do its magic because it would no longer be in its optimum range. But what is going to bring about these changes? Why does the pH range have to be so narrow? Why do I actually have to make 2 proteins? My body has instructions for 2 things that do the same thing. They denature, but why?
Wait, but I think you still end up having the same peaks. Are they different kinds of amylase?
Yes, there are 2 different kinds of amylase. There are 100% 2 different kinds of amylase. My body has instructions to make 2 amylases. And I think it's crazy that my body works this hard. But the reason my body has to work this hard is because they have to live in different pH environments.
It knows that the mouth is not made of stomach acid. It's not gonna work that way.
How do they know that?
How does it know that?
I think it's sentient.
God, I think every molecule might be sentient, Okay, so I want to go back to this picture of tertiary structure, and I want to look at this interaction between arginine and glutamic acid. Lo, I have taken amylase from my mouth and put it into my pancreas. And so what has happened to these 2 thingies? I want to say moieties, but you don't like that word, so thingies. So I have taken something that should be at a pH of 7, and I have put it into my, okay, the bond breaks, the ionic bond is going to go away, but I can be even more specific than that. I can tell you that I'm going to protonate that. I am going to functionally change the side groups. I am going to go through it. Anytime I used to have a minus charge, I'm going to be able to put a hydrogen there. And anytime that I used to have an OH, it's completely reasonable at some pH that I could do that to tyrosine. But when I do that to tyrosine, the tyrosine might not be so in love with this histidine down here. Histidine we associate with a positive charge with the histones. I don't know. So the reason that this happens is because the tertiary structure is broken. As R groups are protonated or deprotonated, we're going to break ionic bonds. I don't know, we're going to mess it up. That's why it denatures, because you broke it.
Is that because, wait, is more H plus more acidic or
more H plus is going to put it in a more acidic environment.
So is that because, is it get proteinated because the more H pluses in the acidic environment like bond to the thingy?
Yes. And the same thing happens in base. We suckle off all the H pluses, so we're going to have the inability to do this. So these curves are symmetric because we don't really have a strong sense of positive and negative or more protons or fewer protons being meaningful. But we can also think about how you cook an egg. So you can ceviche some fish if you're in the mood. Ceviche is taking raw fish and putting it in lime juice and trying to cook it using the acid in the lime juice. So if you go to a Mexican restaurant, you can usually get like a first course that's ceviche with some fresh cut vegetables and some fish and some lime juice. Okay, that's fair. But like I also believe that some of the interactions in this amylase are different. I think they are meant, like it folds based on the fact that the pH is lower, that there are more things proteinated. So I think if you went through the primary structure, you would find differences, and if you and I understood more about that, we might understand like how the folding occurs or how it differs between those two structures, given that sometimes we can make an ionic bond in one and we can't make that same interaction in another. I don't know, you could overlay them. You could be a biologist if you wanted. It's too much? Yeah, life is hard. So now we're gonna cook an egg or fish or whatever it is you wanna cook. There you can make cheese, right? You can take milk and add lemon juice and make curds and whey. That is denaturing the milk protein. You like put it in cheesecloth, so you like collect the curds and put it in cheesecloth and press it. Voila, paneer. Do you make paneer ever? You make paneer? Yeah.
What is tofu?
Soy milk cheese.
Soy.
This soybean plant. So here's our cooking our egg. I'm gonna get through this. The egg is meant to be at about 40 degrees because I guess chicken's at about 40 degrees. If you cool that chicken right down, you will slow down every metabolic process in that chicken. You might kill it in the process, but you know that you can take a goldfish and put it in liquid nitrogen. You could do that when I was young. Do you know that? That was a demo that science teachers could do. You take a goldfish, you throw it in liquid nitrogen, you freeze it, then you scoop it out, put it back in water, and it would come back to life. Like its outcome wasn't great. It didn't last for a long time, don't get me wrong, but it would start swimming. And we could do things like that when I was in school because we didn't understand animals. Yeah, we were doing some harm. Like I said, the goldfish didn't have a long, happy life afterwards. It had a short, happy life. I'm not sure it made it home from school that night. Like,
Wait, I'm confused. So it was just like a couple of explanation. You just got the goldfish out in water.
Nobody felt bad?
it came back to life. Like, I mean, Walt Disney's brain is sitting there on ice. Like, this is what we've done. We've changed all that. Yes. I thought it was his head or his brain.
What?
Head.
Yeah.
Yeah, it's like.
Have you guys done cryotherapy for sports?
Yeah, like ice baths. They suck. But I know they freeze Walt Disney's body.
They freeze whose body?
Walt Disney.
Oh, his head, I think.
Albert Einstein.
I believe it. I don't know. I don't know where conspiracy theory like starts and where like people got too much money and did stupid things ends, but like there's definitely an intersection.
It's like they just wanted to wait until we had the technology to bring them back.
So these people are sitting over here trying to slow down their enzymatic processes, trying to like make that brain last forever.
You can pay for it.
That's what I'm can draw both of the curves. You can draw both of the curves.
Bro, that's just, but for using for 70 years,
Okay, so most of you in the room know this answer. I'm just trying to get you to say that if you heat up a protein too much, it denatures permanently. So if you cook an egg, it is unlikely you're going to uncook it. There's some YouTube videos showing you some fantastical things that have happened in this world. But like on the whole, this is unforgivable. The idea is that as we put more kinetic energy in there, the potential energy of the bonds that we had in tertiary structure just ain't worth it anymore. I got so much energy, I can just fly off and do my own thing. I can overcome that potential energy interaction, but it is essentially irreversible. Another thing that is irreversible is that heavy metals can target cysteine residues. Cysteine has that sulfur in it. The sulfur has some lone pairs on it. The lone pairs on the sulfur can make coordinate covalent bonds to the metal.
Is cysteine a protein?
Cysteine is an amino acid with a sulfur. It's the same one that made my hair not curly, my lack of it, but your hair curly because you've got some. But that's like if you're living in Flint, Michigan and you're wondering why your kids aren't developing properly, it's because the heavy metal, the lead in the pipes affected their ability to make some proteins. Like they didn't fold right. And so when the proteins don't fold right, like you don't work the way you're supposed to work. Are Andrew? I kind of do know that because I think they had some relationship to Deerfield Academy. Deerfield's like a fancy private school that my high school friends' parents taught at before I knew her. Deep dive there, Audrey, deep dive.
What about, well, how are they related?
I think that Andrew's parents had some association with Deerfield Academy. It is a school that like I knew about from my own youth. I went to a pretty fancy, pretty old private school in LA, so she was a teacher at that school.
What year was your yearbook things that we can apply to? That was
1990, I was the 101st graduating class. You want to know what they did for the 100th graduating class? Everything. Want to know what they did for us? Woo!
What do you mean by like everything?
They had like all sorts of people back and talks and fancy things and parties. It was an all-girls school. Teas.
That was my next question.
Marlboro with an UGG at the end.
So not the cigarette.
Marlboro like the cigarette with an UGG at the end, like the Duke of.
Marlboro.
The Duke of Marlboro? Here's some questions you can try to answer. Again, that's why I chose this set, because the other one didn't have any fun questions. Do you know only one person has done the homework? Let's do Friday, like don't get me wrong. It's just like the girl who most overtly doesn't like this is the girl that did it.
I turned it in.
That's what I mean. Like, yes, I saw you guys talking about it. I know some of you have done it, but it was still funny to me. Like, oh, state the structural formula of cysteine in a solution at a pH of 3, 5, and 7. The isoelectric point of cysteine, I'm guessing, is 5.1.
Yes.
So cysteine is this, ready to make that disulfide linkage, lose this proton, be reduced. So I draw the Cu, I draw the NH2, and this is when I say, well, in a solution that's 5.1 at its isoelectronic point, I know that this is going to move over here. So at the isoelectronic point, I draw the glory that is the zwitterion. He's looking at the loops of Henley. I remember the loops of Henley. There's a street called Henley Loop just outside of Austin, I think.
I just forgot.
You just drive by and you're like, how is this possible? 1990s.
I think you think I don't. Oh, okay, I don't know.
If we put this in a more acidic environment, that would be a pH of 3. The next proton is going to come and it's going to add here. And so we're going to have that cation form. But if we put it in a basic environment, not only are we going to take this proton, but also we're going to take this proton and we're going to have that anionic form. So add the isoelectronic point, I draw the glory that is the zwitterion. He's looking at the loops of Henley. I remember the loops of Henley. There's a street called Henley Loop just outside of Austin, I think. I just forgot.
I just forgot.
differences? This is a dipeptide. This looks like methionine because it's got a sulfur. This is alanine because everyone knows it's my favorite. What is the other dipeptide that could be formed? So this is methionine, and this is alanine. And the question is, what is the other dipeptide that can be formed from these two amino acids? I heard Audrey say it. I didn't know what she said, but then like
We just switch them.
I heard you, but then I processed it. So she's just saying that we could put them in the other order.
You don't have to say what the peptide is called.
So deduce the structure of the other dipeptide. I mean,
You just say peptide bond between the alanine and the methionine.
explain how a cysteine residue in one polypeptide is able to link to a cysteine in another one. There I am with the R group of the other cysteine that's off my page. What's gonna happen is we're gonna make a bond between these two sulfurs, and we're gonna release 2 protons and 2 electrons. Explain why doubling of, oh please, you're all thinking it.
Yeah.